Hello, I'm Mark Richards and this is going to be a presentation on human immunity. This presentation will cover certain aspects of human immunity and how the body reacts to different immune responses. The main areas covered will be non-specific defence, specific immunity and immune response, the biology of vaccinations, immune sensitivity and health implications. Each section will be broken down to discuss specific areas involved in each. Firstly, we'll be looking at non-specific defences. There will be analysis of how the skin acts as a barrier to infection and the role of inflammation, discussion on the effectiveness of barriers to infection via body orifices, explanation of the roles of the thymus, lymph nodes and lymphatic ducts, and discussion on the role of macrophages in removing pathogens or inert particles. Firstly, we're going to look at how the skin acts as a barrier to infection and the role of inflammation. As we know, the skin is the body's largest organ. It is also the most effective first line defence against infection as a mechanical waterproof barrier. The physical structure of the skin, shown here, prevents most pathogens from harming the body. The epidermis, which is the uppermost layer of the skin that we can see here, acts as a barrier by preventing or impeding incursion of microbes, chemicals, toxins. It absorbs solar and ionised radiation and helps prevent water loss. And as previously mentioned, it is the body's first line of defence, but if the skin becomes compromised and gets broken or cut, it loses that defence, the ability to defend as well. As part of a response to a break in the body's first line of defence in the skin, there may be something called inflammation, and it's usually shown as injury or infection within the skin, and it's going to trigger an inflammatory response. Primary effects of an inflammatory response are increased blood flow to the affected area. Cell walls in the area develop gaps, increasing that immune response. Numerous cells involved in this response are sent to the area along with proteins to help fuel it. Main symptoms of inflammation are red coloration due to the increased blood flow, swollen tissue again due to increased blood flow. It could be painful due to the inflammation increasing the pressure down below on the nerve cells in the skin, and this inflammatory response will continue until the infection or injury ends. And then we can move on to looking at barriers to infection via body orifices. And as we previously discussed, the skin acts as a barrier when it is not broken or compromised, but body orifices provide direct entry for pathogens to enter the body. These are the gastrointestinal tract, which runs from the mouth to the anus, the respiratory tract, which again is all to do with the nose and the mouth, the urogenital tract, which obviously is to do with penises and vagina, and the vaginal tract furthermore. Typically, all these have a mucous membrane coating to fight foreign incursions. The gastrointestinal tract also has an acidic environment, enzymes, bile and intestinal secretions, helping destroy most pathogens except for typhoid and cholera. The nasal cavity is where most pathogens will enter our bodies, but they will mostly get stuck in the mucous lining within the nose, and this is usually cleared through sneezing or coughing. The urinary tract is another way that bacteria can find its way into our bodies. The urethra in males, though, is a lot longer than it is for a female, and it acts as a barrier alone in that it's too far for bacteria to travel up. But in females, because it is a fair bit shorter, bacteria finds it's a lot easier to travel into a bladder. So for that, passing urine helps flush it. Really, that's the best defence that we have to stop this bacteria incursion. The vaginal tract again contains bacteria and to help keep bad bacteria at bay, and it's also a very acidic environment. We move on to the role of the lymphatic system in our immune response. And the thymus is a gland which is just located here, and it's a gland of the endocrine and lymphatic system, and it produces hormones, in which some of which will impact on itself, and it helps produce and mature T-cell lymphocytes, which are white blood cells, which in turn get transferred to the bloodstream via the lymphatic ducts, which act as a gateway between the lymphatic and circulatory systems. The thymus changes throughout a life cycle, and as age increases, its ability to produce T-cells decreases, increasing risk of infection. Lymph nodes are the small bean-shaped tissues which we can see dotted all around the body, and these contain lymphocytes and macrophages. These both act on foreign particles that are present in the lymph, or which is the fluid running through the lymphatic system. As we've just mentioned, macrophages, these are hugely important to our immune system. They are white blood cells formed as an immune response to infection or the build-up of damaged or dead cells. They come in different types depending upon their environment, but all carry the same function to search for and destroy pathogens or inert particles in a process called phagocytosis. This is where we can move now on to specific immunity and immune response. Again, we'll be discussion on acquired immunity and sensitivity to antigens, explanation of the roles of different types of immune cells and immunoglobulins, analysis of tolerance to own tissues and the rejection of transplanted organs. So, talk about acquired immunity and sensitivity to antigens. Antigens are the parts of a pathogen that signals to the body that it is under attack, causing an immune response. They can come as any type of molecule, so proteins, carbohydrates, nucleic acids or fats. Acquired immunity is as basic as it sounds. It is immunity that wasn't present when we are born. Essentially, it's a learned response to our environment and how our bodies learn to attack the antigens and pathogens and create a memory of how to do it. Artificially acquired immunity comes inactively where a person is intentionally exposed to an antigen via a vaccine, and passive where preformed antibodies are introduced, so that'll be antibodies grown outside of the body but introduced into a person. Sensitivity to antigens can be an issue with immune response. Hypersensitivity is a pathologic immune reaction caused by immune response to repeated antigen exposures. This is seen in autoimmune diseases. Moving on again, onto immune cells and immunoglobulins. We have B cells and T cells in our immune system. T cells make up around 80% of lymphocytes and can be split into three groups. We have cytotoxic, which defend against viruses, reject tissue grafts, but rely on helper cells. These work by punching holes in the infected cells. Suppressor cells suppress other T cells to regulate other immune system cells and helps prevent autoimmune attack. And the helper cell, which is the most common T cell in our bodies, making up around 75%, aids our immune system, regulates immune function, acts through the release of lymphokines, stimulating other T cells to grow, attack, and also aids B cells to reaching maturity. B cells make up 10 to 15% of lymphocytes. They mature in the bone marrows. They produce the antibodies that we use to fight infection. They present antigens to the T cells and signal T cells to activate. Immunoglobulins, or antibodies, have the main functions to target infection. They will recruit immune cells, neutralize toxins, remove antigens. They complement activation, stimulate foreign substance, destroying cells, activate mast cells, and can be categorized into G, A, M, E and D, which each have a different response to different catalysts. Now again, as we move on to immune response and tolerance to our own tissues and organ rejection. The immune system has a key task in recognising which antigens belong to the host body, thus stopping its immune system from attacking itself. If confronted with an unrecognised antigen, an immune response will occur. When transplants occur, or transferring cells, tissue or organs from a donor to a host, the immune system can play a huge role as a barrier, as it recognises the donor parts to be a foreign object, attacking them parts and instigates what it deems to be the correct response. Rejection happens in three ways. It can happen hyperacutely, which can be within minutes of a transplant, and it's where the antigens are completely mismatched, so the body just recognises it as a complete foreign body and attacks it with its immune response. Acutely can occur from week 1 to within 3 months. Most patients will suffer this kind of rejection, and then chronic can go on for years and years. Immune response, it just lasts. It's just so slow that it slowly damages the transplanted tissues or organs. Moving on to the biology of vaccination. Well, again, the explanation of the actions of vaccines that provide active and passive immunity. Analysis of scientific factors influencing immunity provided by a vaccine. Analysis of factors influencing vaccine safety. Discussion of the concept of herd immunity and isolation of infected individuals to protect communities. And evaluation of the effectiveness of 3 vaccines: flu vaccine, the MMR vaccine, and the hepatitis B vaccine. So firstly, we will look at active and passive immunity. Now, active immunity can come in 2 forms: natural, which develops from exposure to pathogens and results result of body's immune response, and the learned antibody response if a pathogen is re-encountered further down the line; or artificial, where an individual is exposed intentionally to antigens to generate immune response. This is what we get through vaccinations. Passive immunity also occurs naturally, where antibodies are transferred person to person through natural means, which is like what happens between a mother and her baby. And artificially is where a person is injected with antibodies made by somebody else. These work in a traditional sense, but is short-term solution as they degrade, and as the host immune system is not involved in any way, they are not replaced. Then, as we're moving on to factors influencing immunity from vaccines. Many factors influencing vaccines and immunity they provide. Age is one such factor. Vaccine-induced antibodies decline massively in elderly people, meaning immunity response declines with it. Comorbidity, where vaccines are less effective in people whose immune systems is already under stress from being overwhelmed by having already infections or diseases that it's currently fighting. Immune response falls if it has numerous fights to perform. Vaccines are given in these instances, but the effects, again, will be less. The time since a vaccination. Immunity from vaccinations can fall depending on what the vaccination is for. Lifelong immunity is rare, very rare, which is why we have booster shots throughout our lives. These are recommended at different times throughout our life. Again, other scientific factors that come in to vaccines are how the form of delivery is. So, are they subcutaneous? Are they intramuscular? Are they oral? And also, is the vaccine a live vaccine or an inactive vaccine? Also, if the vaccine does not match a prevalent strain, like it would, for example, the flu vaccine is usually given to what they expect to be the most prevalent strain that year. If it's not going to be that, then you've been vaccinated in theory against something that you're never going to get. So if it's not the prevalent strain, then its performance is going to be severely limited. Vaccine safety. Now, we've all seen in news about people who've been entered into clinical trials for certain vaccines, and there's been accidents. Now, vaccine safety is an integral part. There are robust and rigorous standards and testing companies must adhere to. There is a two-part stage with laboratory testing that contains three things: of a preclinical assessment, we can see on there. If this is deemed successful, they will move on to clinical trial, where people are tested, and it will be a small group of people that are tested. If again this is successful, they'll move on to phase 2, where again it's tested on more people to see what the effects are. It can be a couple of hundred people. If again it passes this stage, it will go on to a further clinical trial, which is a larger trial where it's rolled out along a larger sample of the population to see what the effects are in a larger group. If there's any failure in any of these trials, any of these three phases, the vaccine and everything is ended. Constant monitoring though happens after it's been given its license, because there's no it can't be ensured that the population that the vaccine has been tested on won't have suffered side effects, and that people that may be given it that wasn't tested on may suffer totally different side effects to the population that they tested. Herd immunity. Herd immunity is the concept of which a population is immunised against a certain disease, that if someone caught it, the disease wouldn't be able to spread due to the immunity built up around it. The proportion of immunised population needed for herd immunity differs between each disease. Isolating people who become infected is needed to prevent further transmission. This process is called protective isolation and is imperative for people suffering from highly contagious diseases. These isolations again vary in length by disease. Looking at the three different vaccines that we have here, we've got the flu vaccine, and again, like we previously discussed, its effectiveness depends upon who is receiving it and what strain it is for, whether it is going to be for the prevalent strain that year. But even that, the flu vaccine is beneficial, as it can prevent catching flu, reducing the risk of connected hospitalisation from flu, aid people with other chronic health issues that flu may affect, and it's effective for women who are or just after pregnancy and can pass it to baby through exchange, and it also helps build up the idea of herd immunity. The MMR vaccine has been shown to be very effective at preventing people developing measles, mumps and rubella. We've got the stats: 2 doses of an MMR vaccine are 97% effective against measles, 88% effective against the mumps and 99% effective against rubella. Even just 1 dose of MMR, 93% effective against measles, 78% effective against mumps and 97% effective against rubella. Now, we do know that there has been negative press about the MMR vaccine, with links to autism, and I think the stats that we have here, they pretty much talk for themselves. And hepatitis B, it is the complete vaccine series induces protective antibody levels in more than 95% of vaccinated infants, children and young adults. It's been available since 1982, and it has been 95% effective at preventing infection and developing further chronic disease. There's been more than 1 billion doses of the hepatitis B vaccine issued since 1982, which I think it talks for itself. Finally, as we reach the end of this presentation, we'll look at immune sensitivity and ill health. We'll discuss how the immune system may cause ill health and the ways of reducing risk of allergy. Now, our immune systems are designed to protect us from infections and diseases. Some people may suffer from their own immune system attacking their healthy cells within the body. This is called an autoimmune disease. And as we can see here, these are the different types of autoimmune disease and the different areas that they will affect. There are over 80 different autoimmune diseases, with a lot of them sharing the very similar characteristics, making it hard to sometimes distinguish between each one, which can lead to problems in diagnosis. Initial symptoms are usually fatigue, muscle soreness, low-grade fevers. There could also be inflammation within the body. This is distinguished by redness, heat, pain and swelling, as we've previously discussed. The most common areas that autoimmune diseases are affecting are joints, muscles, skin, red blood cells, blood vessels, connective tissues and endocrine glands, with diagnosis only coming by identifying what antibodies are prevalent in the blood at the time of a blood test. And when we come to allergy, allergies are reactions by the immune system to certain substances which can seem harmless to most people, but they are affecting around 25% of the UK population at some point in their lives, and this figure is only increasing year on year, with more younger children becoming more susceptible to allergies. They can come in all sorts of different areas: foods, dust in the house, moulds, pets and pollen, simple hay fever. But they're all quite regular in our households, that we have in one form or another. Reducing the risks are quite simple. In food allergies, reading your labels to make sure that you're not eating something that you know you are allergic to. Steering clear of that catalyst. Wearing an alert ID if you're eating out, so people know that you have this allergy. And in times of severe reactions, being prescribed an EpiPen for epinephrine, which is an initial first defence shot of adrenaline into your system. In the household, with pets and dust, keep in contact to particular animals that can cause the reactions to a minimum. Washing your pets regularly, clearing all excess fur, cleaning litter trees, as it's usually the skin, dead skin cells and the urine, dried urine, that is the actual catalyst for the allergy. And with dust mites, again, it's a common cause in the household, and having easy clean floors, easy clean blinds, easy clean, removing upholstered sofas and going for leather sofas, simple things like that. Cleaning upholstery regularly if it's fabric, using a HEPA filtered vacuum cleaner. They're all simple things that can be done. Taking your regular medications, if you're prescribed medications for things like hay fever, making sure you're taking antihistamines to stop that allergy response. So this is the end of the presentation, and we've looked at the non-specific defences and broken that down into its component parts. We've looked at specific immunity and the immune response. We've looked at the biology of vaccinations. We've looked at immune sensitivity and health implications, and each section was broken down to discuss specific areas involved in each. Thank you.